Good morning, everybody. I hope you can all hear us well. Welcome to the another wonderful Zoo and Wildlife Study Group lecture. My name is Perry Wolf, and I am the Executive Manager for Wildlife Disease Association. And I want to go ahead and introduce you to Alejandro Castro, who is the leader of the Zoo and Wildlife Study Group, and he will introduce our speaker.
Thank you. Okay, so as you know, like we've been doing this for several months already with collaboration with the WDA. And in this opportunity, we have Dr. Santiago Jiménez. I had the opportunity to like be in one of his lectures while I was in veterinary school, like several years ago. And today he accepted the invitation to join us today. So Dr. Jimenez obtained his veterinary degree from the National University of Colombia in 2003. He conducted research in wild felids, anesthesia, hematology, and medicine in several species, including tigers, lions, jaguars, pumas. And after graduation, he worked in several institutions in Colombia. And after that, he moved to the United States, where he now works as the small animal practice representative for the Maryland Veterinary Medical Association. He also did an internship at the Smithsonian National Zoo, where he was doing reproductive science with emphasis in clouded leopards. So Dr. Santiago, welcome. And again, thank you for accepting this invitation.
Well, thank you. Thank you very much for the invitation. I'm really happy to be here. I just want to share some of my experiences in working with big cats. This has been a really, really fun journey, long and really hard. And I could not accomplish anything that I've done with the help of many people. So this is a collaborative effort from like multiple institutions and friends, family, like everybody has been involved in this. So the purpose of this talk today is to share these experiences. I'm not going to go in detail with like hard science. I'm going to share some of the PDFs and the publications that were the product of this research, so that you can look into the numbers and the more in-depth results. So I'm going to start sharing my screen, and we're going to start with, well, this project that I'm presenting to you today was kind of like part of my PhD project, and was titled Demographic, Genetic, and Reproductive Analysis of Captive Jaguars. My director was Dr. Claudia Jiménez-Escobar in the National University of Colombia. And but I want to share with you a little bit of my background. So before doing this study, as Alejandro mentioned, I did some research on anesthesia, hematology, and retrovirus diagnosis in 5 species of captive felids in Colombia. And I did that with my colleague, Wilson Moreno. So basically we work with different species of cats, lions, tigers, jaguars, pumas, and ocelots. We perform anesthesia on many of those cats. And the idea was to understand a little bit better how the anesthesia was working, and there was some like differences between the anesthesia protocols between the species, and gathering some values for hematological reference values, and also take advantage of those procedures to diagnose different diseases like the leukemia virus and the immunodeficiency virus. All the cats that we tested at that time were negative for those. Only one lion came back positive for FIV, or the lion lentivirus. So that was kind of like some of the exciting part about that. And after doing this project, I wanted to concentrate more of my efforts on working with jaguars. So that was part of the main reasons why I started like this new project, and I started working as the director in chief for one of the NGOs that led the first conservation plan for big cats in Colombia. So one of the problems that we found at that moment was that jaguars were really threatened, and that they were losing their habitat, and there was so many threats. So it was a necessity to work with jaguars. So, there's two presentations of jaguars, the melanistic jaguar and the spotted jaguars. And we had many in captivity in Colombia, so it was a very good population to study. So, and of course, the main problem that I faced at that time when I was organizing the project was to see that, that we were losing them, and that the habitat was getting lost, and that there was a necessity of doing something about it in terms of like conservation. But we know that conservation is something that is better to be done, or preserve the species in the natural habitat. But we had a big collection of captive animals that are very important to study, so then we can help the animals that are in the wild. So one of the things that I noticed was that we had a lot of jaguars, but we didn't know a lot about them. We didn't know how the population was composed and how were they breeding, and so we have a lot of gaps in this family tree. So one of the first aspects that I wanted to achieve was to organize those populations. So here in this slide, we see like the geometrical figures are like different zoological parks. The yellow dots are just like spotted jaguars. The black ones are melanistic jaguars, males and females. So we had this big population, but we didn't know a lot about them. We had some records and we had some information, but we needed to clarify that. And also I saw, for example, that one of those cubs that were born at that time was born with cleft palate, like a teratology, like a really congenital problem. And that makes me wonder, like, okay, what's wrong with this? Is there some genetic things here, or is there any inbreeding that is causing these problems, or what's going on here? So I started to organize like the information for the project, and the first step that I wanted to do was the demographic analysis. But probably people that works on demographic and stud books and all of that, well, we know that when we don't have good records, then we cannot get good values. So, and that was the first challenge that I faced. And it was like, there was some lack of information. So it was hard to compile all the information. So we had all those gaps, and that made me just go back to step back a little bit and organize a genetic analysis, so then we could understand a little bit better the composition of the population. So I have to review like all the literature. That was not, there was not a lot of studies there, but probably there's a couple of ones that is important to mention, like the studies made by Dr. Ruiz Garcia and Esteban Payan. In Colombia, they analyzed and they characterize like all the population with different macrosatellites, and they understood a little bit better the genetics of the Colombian population and the Latin American population. Isaac and the group of Dr. Stephen O'Brien and Warren Johnson, they also did their own study, and I used those studies as templates. Now, when I study those papers and everything, okay, I didn't understand a lot because it was really complex. So my purpose was not to become a geneticist, but just to use genetic tools to solve some questions. So basically what I did was just, okay, let's go and collect samples from the jaguars, the captive populations. It could be like hair samples or blood samples. And in the case of the blood samples, we isolated the leukocytes, extracted the DNA from those leukocytes, and run some microsatellites. And the microsatellites are just for the people that doesn't really know what it is, it's just like sections of the DNA that doesn't codify for anything. It's more like the structural DNA. but can have some variations over time and between individuals and between populations. So they're good markers just to understand how different are one animal from another, and they're used for paternity tests and maternity tests. So now, ideally, we wanted to do also analysis of the mitochondrial DNA to understand better the maternal lineage, and also Y chromosome analysis to understand better from the perspective of the paternal side. So, but based on like the resources and everything, it was really hard to achieve all of that. So we just selected a panel of 9 microsatellites, and we obtained some genetic profiles of these individuals. The idea was to analyze that and establish and build the pedigree of the population. So at that time, I worked with like 20 jaguars. There was like 36 captive jaguars, so I was able to collect samples from 20 of them. And there was some that I got blood. One individual I got cartilage because it was a skeleton, and the other ones were blood and hair. This is just an example, a short video to show how I collected some of those hair samples. And there's other species here, but the idea is that I'm going to highlight a couple of points here. So this was a melanistic jaguar. Her name was Milena, was in the Santa Cruz Zoological Foundation. A very beautiful animal. So the idea was to try to do also non-invasive work. It was like, okay, let's try to collect these samples without anesthetizing the animals. So, well, I used like some leaves just to tease them and scratch them. So then try to make them get close to the fence and try to collect the sample from the hair that I could get. So that was an interesting experience. Of course, we need to be careful. You're going to see a couple of things that we should not do when we are collecting these kind of samples. This is another jaguar. They just like to scratch in the fence, you know. So I use that leaf just to tease them, and then try to collect the sample with a hemostat. So that was fun. Well, this is the part that you should never do. You don't put your fingers inside the cages. That's something that is very important. So we've all done that, but we should not do that. So now this is like other cats that I collected samples as an example. Of course, the samples that were analyzed were the jaguars. So, and it's a tiger, and the next one is going to be a really special cat. It's a liger, which was the only animal that was in the collection. That those, you know, for ligers, nobody encourages to breed lions and tigers, but this happened at some point. So that animal was there. And it was also interesting just to see what was happening genetically with this animal, but it's not included in this study. Now, for the samples that were more advanced, like in terms of like blood samples, we anesthetized the animals, and of course there was some like a purpose. For example, those animals needed a routine checkup. In this case, this was in the Matecaña Zoological Park, close to Pereira, and I would like to thank Dr. Delio Orjuela and Angelica Moreno just to help that day. We worked with 5 jaguars the same day. So they were just like changing animals from one cage to another. So we took advantage to perform the procedures. So this is just an example. So we put the dart, and you've done that many times. So we waited until the animal was like anesthetized, so then it would be safe to work with her. And the dosages of the anesthetic that I used were kind of like the ranges that I obtained in my previous study. So we were comfortable doing those protocols. We used like xylazine and ketamine at that time. And later on, we can do another seminar just to talk just exclusively about anesthesia. So we collected the blood samples, and we did that in different zoological parks. So it was a very good experience. Collected hair as well. Actually, in some animals, we collected feces. So we also did extractions from feces. like DNA extractions from feces. But of course, the best samples that we can work with were the blood samples. Now, I also tracked some individuals that were in the natural history museums, and we found like those animals that could have been the founders of the population. So it was the cats that you see here in the slide. Those are the cats that were alive in the 70s, 60s, and 70s. So I tracked them down. I collected samples. Unfortunately, the chemicals used to work on those first, they ruined part of the DNA. So we didn't get amplification for those, but we got DNA from the cartilage of this skeleton. So that was very, very, very important. Now, in terms of the genetics, we selected different primers. Those are the microsatellites that I was mentioning, and they are labeled with different colors. So then we could see them during the electrophoresis, and that's the technique just to visualize those peaks. And those are the sequences that frame those sections of microsatellites. So we selected 9, and then we perform like PCRs. So this is kind of like the conditions for the PCR that we use at that time. So that's the polymerase chain reaction, just to amplify the DNA and just create like millions of copies of that. So then we could have seen that, we could see that and analyze that. And my apologies if you're gonna see a lot of Spanish things in Spanish. So, and it's gonna be, it's part of how the presentation was done. And then after obtaining the samples, then we needed to analyze that, and we used different softwares. And that was like the first part was made with Sparks. Sparks was just to build the stud book of the population. Then that was exported to the software PM2000. And that can give us more like the population analysis and all the rates. We use GenePop to analyze the microsatellites and establish like other genetic measures like the Hardy-Weinberg equilibrium and other measurements that are very important, the genetic diversity and all of that. We use kinship, and this was a software that established relationship between individuals. So the idea was to know if those individuals were related to each other. And we use service, and this software is designed usually just to assign paternities and maternities. So we use a lot of software just to analyze this. Now this slide shows like the genotypes once we did the PCRs and we amplify those and we got the electrophoresis for each individual, and for each primer we got like different alleles, like a maternal and a paternal allele. So we're gonna see like the genotypes of all these Jaguars, and unfortunately for some of those microsatellites, they didn't amplify in some areas. So we got those as missing data. And we established that the allelic frequencies, and that's important. I'm gonna show you why. The purpose of that is just to understand what microsatellites are more polymorphic. So the more polymorphic the microsatellite, the better we can like establish like the diversity of and the relationship between individuals. So for example, for a paternity test, we don't want to select this one because it's not highly polymorphic, but we want probably to select the 008 because it has different alleles. So it will give us a better information. So now when we run the program, the kinship program, we found that with different levels of significance, we found that there was some relationship between individuals. Like let's say the first couples here were close with a 95% probability, and 3 pairs with 99%, and 11 pairs with 99.9%. So that's what is really important, though, that these animals were really related to each other. Now that we needed to find out how. So we use service to assign those paternities and maternities, and that selects like usually like confidence intervals, and there's a flexible and a strict one. So we got kind of like the assignation for that. Now, this is an example of how the genetic sheet looks for each individual for primers. This is just a summary of that for these two individuals. And then we did kind of like, we did paternity test. It's, for example, here in this slide, we see that this is the genotype of one individual. We knew the mother, and so we found some of those alleles there, and we needed to find the father, and we found it, the one that just matched and complete like the genotype of the offspring. So that way, this was kind of like one of our controls because we knew about this triplet. So then going back to this family tree, so we had all these individuals, and we needed to start just like, okay, how these animals are related to each other. So the first step was to build a stud book, and we analyze the records, and then that give us kind of like this information saying, okay, from these two parents, they have this animal that was transferred to this zoo and all of that. So we established some relationships with the stud book, but there was some animals that we didn't have any clue. We didn't know anything about them. So that's when we did all this genetic analysis. Now, when we established the kinship, when we analyzed the information by kinship, we established that there were some relationships between those individuals. But it was necessary to do the service because that was the one that was assigning the paternities and the maternities. So then we ended up with something like this, a little bit more robust, like something that was telling us like, okay, from these individuals, then we know that this is the offspring of these two individuals, or this one, or this melanistic jaguar, with different levels of confidence. So that was kind of like the outcome of that. Now, knowing this, that was when it was important to put that information in the stud book again. So then we can run all the population management analysis. Now we had more information, and definitely the microsatellites and the genetic analysis and the assignation of paternities and maternities helped us to make that information more robust. So once we did that, then we obtained like different genetic values, statistics for the founders. So it was, we're starting to be able to analyze this information. So this is just an example about the founders and the descendants and the contribution from those founders to the next generation, the allele retention, and the potential for those allele retention. And we see like, we got this information about the genetic diversity. So like close to 94, 95, we had like a 50% genetic diversity. It was like a really in the middle. But there was a moment where the animals in the captive population, there was a boom in reproduction. Like every single zoological park started having like cubs, and they had the space for those cubs, and that increased dramatically the genetic diversity of that population. So the breeding programs that were established at that time in the Colombian zoos improved the genetic diversity, rising those two levels to 0.78. So we had a 78% genetic diversity in the 2000s. So definitely that breeding process contributed. But also, we found that in that same process, the inbreeding also increased. So we found that the inbreeding coefficient, that's like what was proposed by Wright and other authors, it was 0. But when the population started breeding, it increased. It was different than 0. It's a low inbreeding coefficient still, but it was different than 0. So somebody messed up here. Like somebody bred some individuals that were not directly related, but for example, like first cousins or second cousins, those kind of things that just can change that inbreeding coefficient. So then we saw that, and then got stable because they were not breeding anymore. So all this study was published in a book. It was a chapter of a book called Big Cats. I'm gonna send a copy of those papers to Alejandro, so then they can be distributed if you want, or you can find them online. They have free access. You can look for my name in the ResearchGate webpage, and then you can find all those PDFs there. So that's going to be, that was kind of the first part. So we understood the composition of the population, and we had better data just to start doing this demographic analysis. So, and in different zoological parks, like especially in the US, they have these species survival plans for different species. And that's probably one of the best efforts for conservation, for ex situ conservation. This is just an example of the Siberian tiger plan, and they establish all the relationship between individuals. That was something that I found like in '93 when I was just a student. So then I just, okay, I want to do something like that with jaguars. Now I found another study that was done, was made by Drury, and it was like investigating like the inbreeding in the European population of jaguars. They found records from the 1920s, and they found that the inbreeding coefficient was changed through time, and it was decreased just due to the introduction of new founders. So the introduction of new animals to the collection always improves the genetic diversity and decreases inbreeding. And the thing is that we don't want to overpopulate the zoological parks. It's just like a consequence of like all the illegal trafficking, all the animals that cannot return to the wild, that unfortunately cannot return to the wild. So somebody needs to take care of them, and that's the main purpose of the zoological parks. Just like to give them refuge to those animals, and also to study them, because thanks to that we can improve and we can help the animals that are in the wild. So, okay, sorry about this one, just Spanish, a lot of variables. So basically we got all the information from the animals, species, registration, microchip, sex, well, gender, reproductive status, and all of that. So all that information was entered in the Sparks software for building the stud book. And I wanted to thank this moment, well, all the people that helped me in that part. I went to Venezuela, to Caracas, and to Panama to take the different courses offered by the International Species Information System. And I got the training from Lori Bingman and Catherine Rodriguez Clark and Kristen Lewis, and they helped me a lot just to organize and start just building this stud book for jaguars. So these were the zoological parks that were involved in the project. So we use Sparks and then PM 2000. I think that you guys are using PMX, and that's a newer version. I think that those things have changed. So basically what we got was just general information about that, like the distribution from males and females. We got the census from 1970s, and all the demographic characteristics, the mating selections based on the mean kinship, and established some population goals for the population. So at that time we had like 44% of the population were males, 56 females, 80% spotted, 20% melanistic. All the females were intact. Some males were not intact. So some males, in that boom of reproduction, somebody made the unwise decision to perform vasectomy on these individuals. And I think that there was a kind of a mistake at the time. But anyways, we established the ratio from males and females. We established this distribution from the individuals based on the class age. And we noticed that there's some moment where the population was really increasing, but the population was kind of like, we can see that it's getting older, and there was no breeding, no reproduction at this point. So that was kind of like, we want to see kind of like a pyramid here, just to have a very robust population, but unfortunately that was not the case. We found that the population was decreasing. So, and that's this value lambda, which means that if it's higher than 1, it means that the population is increasing in size. And when it's lower than that, it's decreasing. So it was decreasing at that moment when we did the analysis. This was the census, by males and females, the total individuals, females in green, males in red. And of course, we see that decrease in the population from the 2000s to 2010. Now for type of birth, like definitely there was not a lot of individuals introduced from the wild. So most of that reproduction happened between individuals that were already captive. And then we got all those population rates, the fecundity rates, the reproductive value, the survivorship, the survival rate for males and females, mortality rates. So interesting to see here is that we got some old jaguars in captivity. And there were some jaguars that reached like 30 years. So based on the records. So that's kind of impressive, you know, like in captivity, the life expectancy of individuals is extended because they have medical care, because they have good nutrition, because they don't have a lot of threats. So those kind of things increases their life expectancy. We did some reproductive projections just to keep a stable number of individuals during the first 5 years, 10 years, and 20 years, 15 years, and 20 years. And based on this, on all those values, and especially the mean kinship value, that's kind of like the relationship between individuals. Not necessarily means that there's some inbreeding between them, just like that there's some relationship between them. And based on those values, we perform some mating selections just to preserve that. And people that are familiar with these population goals, one of those goals is to keep the 90% of the diversity in 100 years. So basically, that tells us, like, if we want to keep a population of 4 individuals, what are the things that we need to do with that population? How many founders needs to enter the population during that time? And of course, if we don't do any reproductive management, well, that genetic diversity is going to decrease dramatically over time, and the population is also going to decrease. Now, when we do reproductive management of these captive populations, then we know that the genetic diversity is going to increase, that this genetic diversity is going to be maintained over time. So these captive animals could be a reservoir for populations that needs new genetics in the wild. Now, and this is a little bit controversial, but the idea is really hard to release carnivores in the wild, and because they need to be born in the wild. They need to learn from the mother to really have all the adaptability and all the skills that they need to survive. So it's always a challenge releasing those individuals or rehabilitate these individuals. But if we think about that, if we have a wild population that is close, that the genetic diversity is getting affected, that we need to introduce individuals, probably it's better to introduce gametes to that population, embryos for healthy individuals that are compatible with this population, because those embryos can be born in the wild, and the mothers can take the time to teach them all the skills for survivorship. So basically, that's one of those things that how we can use the captive population to improve things in the wild. Now, this was published in the journal Alexander von Humboldt. So you can find all the demographic analysis in more detail with all the other values. And of course, this is a dynamic process. Like this was just a snapshot of what was happening in the early 2000s in the captive population. Now things have changed. The population has changed. So, but it can give us kind of a good template to extrapolate to other species, to other individuals, to other populations. Now, the third part of this study was to, okay, now we know what animals compose the population, what animals are related, what animals are not related. So then we know what animals we can select to breed them properly. but then we had the challenge. Okay, now what? How do we do that? Like, how do we understand? How do we apply all these reproductive techniques for the big cats, for the jaguars in this case? So there was a whole challenge. So we didn't have a lot of animals. We didn't have a lot of resources. So we tried to work with what we had at the Santa Cruz Zoological Foundation. We had 3 females and 3 males. And the purpose was to do some longitudinal analysis of hormones, just to start understanding what was happening with the behavioral, with the reproductive status of the population. So in females, we measured estradiol and progestagens. We're going to call it progesterone here, but it's progestagens during 12 months. And also we did a non-invasive monitoring for induced activity. So just after protocols for follicular growth and stimulation of the ovulation using ECG and HCG that has done with multiple species. And in males, we wanted to check testosterone, perform some electroejaculation. Unfortunately, one of the males passed away. So we did an epididymal wash. So we obtained spermatozoids from the epididymis, and we cryopreserve those samples. So in the case of males, so we anesthetize the cats, the jaguars. We perform the electroejaculation based on the protocols described by Dr. Dave Wilt at the Smithsonian National Zoo. So we collected the samples, we analyzed them by microscopy, and just like classified the different abnormalities and the proportion of healthy spermatozoas, and we cryopreserved those samples. So this is kind of like the non-invasive. Well, these samples were collected where the hormones were extracted from feces. So for that, I was accepted, as Alejandro mentioned, at the Smithsonian National Zoo, at the SCBI, to do an internship working with all these techniques, and I had the luck to work with Dr. Joel Howard, Dr. David Wilt, and Dr. Janine Brown, who just taught me all these techniques, and I learned to do this with clouded leopards. And also I want to thank Bernardo Mesa, who was a colleague that introduced me to the people at the Smithsonian. So I was able to learn all these techniques and extract these hormones from the feces. And then I went back to Colombia to apply that with the jaguars. So this is the first part. We did it with males. So we got some peaks, but we didn't get like a lot of like variability in our second male. So what we wanted to do also, if there was any seasonality or any pattern for that, and we didn't find any significant differences between the different types of seasons in Colombia. You know, Colombia is in the tropics, so we don't have like clear winter, spring, summer, and fall. We have like rainy season, less rainy season, then another rainy season, and then another less rainy season. So we compared all these seasons, and we didn't find any significant differences in the testosterone for those 2 males. Now, this is not, of course, if we want to understand this better, we need a bigger population, because this is just a reflection of just 2 individuals. So, and we got also like all these parameters for the multiple ejaculates from these jaguars. So in average, we found that the morphology was normal in 78%, like an average 68%. So it was a good, like normal spermatozoas in those samples. And we compared that with other studies, like the studies made by Morato in Brazil and all of that. They had great experience doing this analysis. So they were kind of very similar. Now for the epididymal wash, we obtained like just 34%, 36% of natural normal spermatozoids. And we found that there was a lot of distal droplets. And that's a normal finding for an epididymal wash, because those spermatozoids are not mature, not fully mature. But it was interesting performing the procedures and establishing the basis for new projects. Now for the females, so we collected samples from a whole year. We extracted the steroids, the fecal steroids, using like all those protocols with ethanol and methanol and all those solvents. And we performed radioimmunoassay for estradiol and ELISA for progesterone. Like, so we followed those hormones of those 3 females. For during a month, we also did a follow-up after applying, injecting these hormonal protocols, just to see if there was some follicular activity and if there was some response to that follicular activity, and if there was some signs of ovulation induction. So this is just, okay, this is like getting hit by a brick with a brick. It's the statistical analysis to measure these hormones during the year. And then we got these results, very interesting. We saw like the blue lines are estradiol. So we saw like estradiol activity during the whole year. And the most interesting part of it is that we found progestagens activity in different parts of the year. And those animals were isolated from other animals. So normally we know that felids in general, they're induced ovulators. So they just ovulate at the time of mating. Not all of them. There are some species that have been found that they're spontaneous ovulators. So we started just seeing clues for that jaguars can ovulate spontaneously. We found in this jaguar, for example, that there was a period of inactivity at the end of the year. In our second female, we found a lot of progestagens like activity during the whole year, and some follicular activity. Of course, this is not like what we wanted to do is just to establish some averages for times. It was not like following day by day, which is like really expensive and it was not easy to achieve. So this is another female where we saw also activity for all of them. So this can just make us think that there could be an association with this, with the spontaneous ovulation in jaguars and the presentation of these peaks of progestagens. And when we did the seasonality analysis, we found that there was some significant differences in times of the year where we did. With the first 3 seasons, we didn't see any differences, but in the last season, there was kind of some low activity, which suggests that there's some period of anestrus, possibly anestrus at that time, like some inactivity at that time. Same thing for the progestagens. At the end of the year, we didn't see a lot of activity. So then we did the protocols with the ECG and the HCG. And now what we found is clearly there was some follicular activity with these peaks of estradiol. But then there's a confusing findings here because we see progesterone or progestagens at this time, but we don't know if this is a preceding corpus luteum or something like that was producing progesterone before applying this, or if it was just a consequence of that. So that was one of the biggest challenges, that it was kind of like a blind study. Like we didn't have major control for the previous exposure to testosterone, to progesterone. And that can be done with some implants of progesterone that some researchers have done. They put some progestagens on the females, and then once the progestagens are acting in the system, that suppresses ovarian activity. So at the moment that they remove the implant, the ovary is sensitive to these hormones. So probably that's a good approach to really have a good assessment of the function of these hormones and to have a better quality of in terms of like the ovulation that are going to happen afterwards. So, but there's a downside for everything. Using progestagens on females that predispose them to develop like a cystic endometrial hyperplasia, and that can decrease also the probabilities for implantation of the embryos. So there's downsides for everything. Now, in this other Jaguar, we applied the protocol here. We saw some progestations, but we could not differentiate that if that was just caused by the ovulation or not. We saw activity, but we could not like really reach some major conclusions from this. So definitely the message for that is that we need more research and we need to work a little bit more on these techniques to understand better. We know that they work. Now we need to do the fine tuning to make it happen well. So then we can have more success in all the artificial inseminations, or like the achievements that Swanson and his group had achieved, and Dr. Regina Paz, they have achieved with the artificial inseminations in Jaguars. So as I said in the beginning, like I could have not done this without help. So I want to thank the Association of Zoological Parks in Colombia, especially the Santa Cruz Zoological Foundation, the International Species Information System, that now has a different acronym, but I want to thank them. They helped me a lot. The Instituto Venezolano de Investigaciones Científicas, IVIC, SCBI, National Zoological Park in the Smithsonian Institution, the National Cancer Institute, Dr. Stephen O'Brien and Warren Johnson also, they gave me a lot of lights about this. DSI Italy, they provided some of the kits for the analysis of the testosterone and progesterone, and the National University of Colombia. And I also want to thank my mother because she is the person that has contributed more to the conservation of wild cats in Colombia. She doesn't know that, but like she has done that. And all the people that helped me with the papers, Consuelo Espinel for her help in translating some of that, the documents, and no, well, everybody that participated in this project, I'm really thankful for that. So the idea is that this was just an overview of what was done. We can go in detail later, and we can organize other webinars. But I just wanted to share kind of like this experience. It's been a fun journey. I'm organizing a new project, so something's gonna come from all of this. So if you have any questions, please feel free to ask questions. If we don't have enough time to answer all the questions, you can write me to this email, and then I'll be really happy to answer any possible questions and share with you this kind of experience.
Okay, great. So thank you so much, Santiago, for this presentation. It was really interesting, very, very useful, actually. So, so for those who have questions, you can use the Q&A section to submit your questions. And there is a couple of questions. So I'm gonna go ahead and ask the first one. So I'm sorry if I mispronounce some of your names. So there is Joshua Sohail from Pakistan. And the question is, how can we decrease cubs born to the lions and tigers starting developing different medical conditions like brittle bones, weak immune systems, and lack of organs? And especially refers to zoo and private breeding centers.
Okay, so that's a very good question, because everything is about responsibility. And I think that these practices are not good practices. Like, we should not breed different species together. There's no purpose of that. People who does that, they have more commercial interests on these things. So definitely should not be encouraged, especially at the zoological community. Like that was something that was done in the past. I think that in the present, nobody's doing that actively. It's more like those private collectors that they have those individuals and they want to do like weird things and crazy things. So, and yeah, I agree. There's like, I had experience also with animals in circuses. Like they don't have any clue what they're doing, and they some of them, they bred individuals that are related to each other, and we saw that in a population in Colombia. There was some circus where they bred mother with daughter, father with daughter, and the offspring was just like really, really abnormal cubs. And I'm publishing that as a case report, because definitely we see that it's a fact that these practices that are just like are outrageous, and they should not happen. Now, now in the wild, we don't see that. Like the only place where a liger can be born theoretically will be in India, in the Gir Park, where there's still an Asian population of lions and some tigers, but they're not gonna. They need the impronta to see each other as the same species, and that doesn't happen in the wild. It's really, really rare. So definitely that's not a practice that should be encouraged. And we need to do it like in a really organized way. The species, they should not breed. They should not crossbreed those species. Alejandro, I think you're mute.
Oh, you're muted.
Oh, sorry. Yeah, I was muted. Okay, so there's another question from a biologist from Venezuela, Ilat Vivas, who works on jaguar population ecology and conservation. So he says, I want to start a project focused on gen flow between population and wild jaguars. I have 3 questions. Number 1, where do you think is the best place to get trained on chemical immobilization for wildlife? Do you want me to read all 3 of them, or do you want me to go?
So yeah, you can read 3 of them.
Okay. The second question is, would you like to help us on our project? And third one of our wild population shows some sort of skin tumors. We think that could be because of inbreeding or because oil exploitation. Because it's in a protected area, but without an effective connectivity with other population, and where the state have an intensive oil exploitation. Do you think these tumors could be caused because of inbreeding or oil contamination?
Got it. Well, greetings for my friends in Venezuela. I had a great experience going to Venezuela and learned a lot of things about jaguars there. So I'm really happy to help if I can in the project that you guys are doing right now. Now, to answer your first question about chemical immobilization, well, I faced the same question when I was a vet student and I needed my degree. And the first part that happened was that when I was a volunteer and I was a student, a veterinarian died during an anesthesia procedure with a liger in Colombia. So the liger woke up and killed the veterinarian. And that was something that, well, first of all, we shouldn't have ligers. Second of all, why did this happen? Nobody really knows exactly what happened, but it happened, and it was kind of like a traumatic experience for the community. So one of the things that I said to myself, okay, I want to learn how to anesthetize big cats. And there was not a lot of teachers to show me how to do it. So most of the things that I did was extrapolating things from small animal medicine, like learn to anesthetize cats, learn to anesthetize dogs, in this case cats, because they're more related. I study a lot of allometric scales and just like how to extrapolate dosages from, it's controversial, but how to extrapolate dosages from a domestic cat to a puma, for example. There are techniques for doing that, and I can provide some references. And basically, what I did was just traveling the different zoos, and the zoo veterinarians, they had some experience, so shared those they shared those experiences with me. I was able to understand a little bit the processes, and I had to read a lot and then try. Because the thing is that, especially with anesthesia with large carnivores, we need to be we need to be accurate in the sense that we need to be in a safe zone where the cat is not going to wake up and where the cat is not going to pass away if the anesthesia goes too deep. So we want to be in that safe zone. So you can have access also, the person that asked this question, to the thesis that I had, and there's a big chapter about anesthesia in these 5 different species of big cats, and that can help you. And the literature, like the Zoo and Wildlife Animal Medicine, you're gonna find some information, and establishing like internship with different zoological parks. I don't think that there's a specific course. If there's one course, then take it if you find it. But I think that it's more like about networking, talk to people, try to do voluntary work in some areas, follow some people, and study a lot, and then you will have success with those anesthesia. Now, to answer the question about the tumors and everything, well, it's hard to know if those problems are caused by inbreeding or if it's something extrinsical, like some contamination or other things. It's important to collect samples, to do histopathological analysis, to understand the nature of these tumors, and see if there's any association with these other things. Later on, you can do a retrospective analysis of this presentation of these tumors to understand this better. But establishing if it's related to like inbreeding or that is going to be hard just with few samples. So, but there's a starting point for everything. So my recommendation is tumors that are found, tumors that need to be extracted, tumors that need to be analyzed by histopathology, get good reports of that, understand the nature of these tumors, and then organize a plan just to prevent it in the future.
Okay, great, thank you. Okay, so there's another question. I think this comes from Claudia Brieva from Colombia. So I know you did some genetic analysis. What are the best DNA markers for paternity studies? And if you can recommend a paper about this.
Okay, so I think that the best analysis for male and paternity tests involves the analysis of the Y chromosome. And by the way, hi, Claudia. She was my professor. We worked together when I was a volunteer in the rehabilitation and rescue unit of the National University. So greetings, Claudia. So just to answer the question, I think that was going back to that, the analysis of the Y chromosome is very important. And now these days, there are other techniques like analysis of the single nucleotide polymorphisms. Those kind of things will help and will make a more robust analysis. The microsatellites they're still functional. We can still use them. And I think that just for that, we just need a comparative analysis of those techniques and a combination of those techniques just to have a better insight of the genetic composition. But if you want to do more like the paternal lineage, the Y chromosome will be essential. And for maternal lineages like mitochondrial DNA.
Okay, great. And next question. Okay, there's one more question. So from Emily Gonzalez from Puerto Rico. Did you make all your wildlife study degrees in Colombia?
Yes, in Colombia. And of course, I did a couple of things, as I mentioned, abroad. But let's say everything started, like I said, my research career started when I was a kid. I love cats. I love the different shapes of cats. So I was really interested about them. So I started studying since I was a kid, and it was kind of my hobby. When I started when I finished high school and I started vet school, then, in vet school, we don't have like a lot of things about cats. We had more in wildlife. Everything was more like horses, cows, traditional animals, dogs, and a little bit of cats. So I study things by myself, like parasitology of the big cats and virology and all the areas. I always study the component for the cats. And then I started volunteering at different zoological parks. Then I did this project to get my degree. So that was like my undergraduate program, like to getting my veterinary degree. The PhD was done in Colombia as well at the National University. And I had the opportunity to do like the international internships at the National Zoo, at the Smithsonian Institution. And as I mentioned, I went to Venezuela to have these courses for stud books in Panama. So that's kind of like like what form in the places, but mainly was in Colombia.
Okay, thank you so much for all your questions, and Santiago for your answers. So I don't see any other questions, so I'm gonna give the word to Dr. Perry. And thank you again, Santiago, for
You're welcome. My pleasure. I hope that this is not the last time. We can do more things. We can do one for just anesthesia and chemical restriction. We can do something for hematology. So as you can see, it's a lot of topics, a lot of things. So definitely we can have some more fun and share more experiences about the big cats later in the future. And as I said, I'm going to send you the copy of the PDFs, and if people are interested, I definitely just write me an email to largecatvet@gmail.com, and I'll send you the papers so then you can read about that. Our colleague in Venezuela, I will send you the thesis is in Spanish, so you can read about like chemical restriction about the cats there.
Thank you so much.
All right, thank you very
Santiago.
much. Thank you, my pleasure.
Okay, so I think we're done with another amazing webinar, and thank you everybody that attended. And this session was recorded, so it will be, we'll have it up somewhere that will let everybody know where it is. So I don't think we've settled on an exact spot yet, but we can always link it to our YouTube channel at WDA for everybody to enjoy. So
Awesome.
great.
Excellent. Thank you.
Thank
Very
you.
much, everyone.
You guys have a good one.
Thank
Thank you
you.
very much.
Bye-bye.
Bye-bye.
Thank you so much.